Right, so with the theme of the Congress being us navigating towards progress, towards a sustainable future in a really unpredictable world, I thought it apt to share some of my postdoc work, which looked at the future-proofing of the use of ivermectin as an endectocide for malaria vector control. So what endectocides are? So they are systemic compounds that are used to treat both internal and external parasites. So these can be used to disrupt the parasite development both internally as well as disrupt vector transmission. So basically how it works is drugs are administered to humans or livestock. Those drugs will be present in the bloodstream, and when a vector comes along and bites and takes a blood meal from those vertebrate hosts, the vector will take up the insecticide and die, as well as any parasites within the bloodstream will also be affected by these compounds. So there's a lot that makes them very useful for vector control. Because of this dual action, it allows us to target a wide range of parasites. And because of their unique selective pressures, there is a chance that it will delay or prevent resistance to these insecticides. There is some suggestion of long-lasting effects. There's some data on ivermectin showing the efficacy remaining up to 7 days post-treatment. This does, of course, come with the caveat that these drugs are only effective for as long as they are present in the bloodstream. So as they metabolize out, their efficacy will reduce, but it's encouraging to see that it's having a longer efficacy in some cases. And there is new technology being produced to create these slow-release formulations in the form of injectables or implants to keep these compounds active in the bloodstream for longer. And finally, they are complementary. They're meant to work with existing tools. It's by no means replacing anything that is working already. So in the case of malaria, we have long-lasting insecticide-treated nets, indoor residual spraying, other larval source management. So this would just add another layer to those tools already in place towards an integrated vector management strategy. So far, ivermectin is by no means a new control. It's been used for many decades in veterinary health, but its use in malaria control is on the rise in popularity because of the increasing insecticide resistance of our malaria vector species, as well as their behavioral changes where they're adapting to all of our indoor-based control tools, and of course the drug resistance in the malaria parasite itself. So ivermectin presents this new novel approach with its unique method of killing the mosquitoes, its complementarity to our existing tools, as well as its excellent safety profile in humans and animals. It is endorsed by the WHO for use in malaria control, and there are a number of trials that have been conducted in Africa. So they have had some varied success, and they are context dependent. So in many cases, they have shown to reduce the mosquito survivorship and density, but like I said, varying results in different regions. Some places very effective, in others, such as Mozambique, less effective. And that makes us wonder, is there something happening with the vector mosquito? Is there a chance of resistance that is popping up, which is leading to this irregular efficacy of this method used in the trials so far? So the current status of ivermectin resistance is there's none detected just yet. There is species-specific susceptibility variation, but our key challenge here is that we have very limited surveillance started to go off, which means we could be missing an emerging resistance. So it's really important here that we conduct this phenotypic monitoring as well as laboratory selection, which leads on to the experiment that I conducted. So here I was trying to artificially select for ivermectin resistance. So we wanted to investigate whether repeated exposure to sublethal concentrations of ivermectin would artificially select out resistance in a highly efficient malaria vector, which is Anopheles funestus. So we have two laboratory strains. One is an insecticide susceptible strain. The second is a pyrethroid resistant strain. So I tested both of these parallel. I exposed them through a standard membrane feeding assay. I can talk a little bit about it at the end. So I gave them a set concentration This was determined by a dose response assay before I started, so this is the LC50 value. I fed the females, sorted the fully fed females out, kept them for 3 days, monitored their mortality, and used those that survived after 3 days to seed the next generation, and then continued this for 7 generations. If you're interested in the data analysis, that's what I did, pretty much asking ChatGPT to help me with some R code, plugging it in to evaluate the effects of the generation, strain, and treatment, as well as comparing our baseline unselected mosquitoes to our 7th generation mosquitoes. Right, so this is the first little set of results. The key findings are that it was a lot of work for some pretty unsexy results. So what happened was there was no significant difference between the 2 different strains, insecticide susceptible or resistant, and also no change over the generations. So the generations run from 0 to 7. There was something strange that happened in the 2nd generation. You can see Fang in red, peach, had really significantly high mortality in the 2nd generation, but this was quite unusual, and there was unusually low death before that. So at that point, I switched to a fresher stock of Ivermectin and continued on the selection. And from that point on, just sort of random biological variation, but no significant trends. And actually, you can see from the shape of the graph, the mortality increased over time. Comparing the baseline unselected mosquitoes to our final 7th generation, again, no significant effect of strain, so between the 2 susceptibilities, and also no difference from the very beginning versus the 7th generation. So I was left a little bit unsatisfied, so I did a follow-up experiment. So here we wanted to look at the comparative survivorship analysis over a longer time. So we were hoping here, because we thought there must be something between this insecticide susceptible colony and the resistant strain. There must be some kind of difference. So here, I fed the mosquitoes in the exact same way as before, but here I gave them 2 concentrations, a low dose and a high dose, and compared that to an untreated control, and I kept them for 15 days and monitored their mortality, and then conducted survival analysis. And it got a little more sexy. At this point, we can see that there's starting to show a split between the 2 different strains. So this graph shows both the high and low dose mashed together. And here you can see that the FUMAS, which is the resistant strain, showing a significantly higher survival compared to the susceptible strain. And if we look at the low dose, a little bit of a wider split between the 2 again. At the high dose, still significantly better survival in the resistant strain, but at a bit of a weaker effect than at the low dose. So this is sort of what we were expecting to see. So while ivermectin may not really drive new resistance in a susceptible mosquito, it certainly can show up existing insecticide-resistant populations, maybe showing a tolerance to it, which may be explaining why we're seeing different results in different parts of Africa with the different insecticide susceptibility of those malaria vector populations. So this has just kind of opened up the can of worms for us. So there's quite a lot that we want to look into. Of course, you always need to see what the wild populations are doing. So these were conducted with laboratory strains. And it's also interesting, well, it's quite important to know the mechanism of this tolerance and what is driving the underlying resistance, as well as conducting some ecologically based studies. So we're interested to know in the face of climate change, what will happen with an increase in temperature? Is that going to drive resistance up? Is it going to change? Yeah, we really don't know, and that's the kind of the future-proofing part of this. I just want to thank everybody, including my postdoc host at the WITS Research Institute for Malaria, and everybody involved there, as well as Carlos from the Bohemia Project. So they are running the ivermectin mass release trials in Kenya and Mozambique. This is my last WITS presentation because I have moved over to the University of Pretoria. So if you do want to find me, please come find me in the One Health building. You can catch me on LinkedIn or by email, and I'm happy to chat about anything mosquitoes. Thanks.
Okay, thank you so much for that talk. Do we have any questions?
Mike.
Thanks. Hi, Ashley. I was wondering, as far as I understand, the way the strategy for endectocides would be is that it would be provided, the ivermectin would be provided to cattle, and as such, your cattle-biting mosquitoes would be taking up the ivermectin, and that's how they would be dosed. Mm-hmm. So why did you do this experiment with funestus, which are very, very people-philic? And are you thinking of doing it with arabiensis, which I think would be the major target for a South African intervention?
Yeah. No, the reason that we did funestus, it's just out of interest because it was my postdoc host's specialty, and we have access to those mosquitoes. So it was purely... the funestus was also because the Bohemia project, where they conducted in Kenya and Mozambique, funestus is the major vector as well, and that was also kind of the reason why. So they were interested to know, is specifically in funestus, is something happening between these two geographic populations?
Thank you so much, Ashley. It's an interesting talk. And just to add on the question to the question from Ushone, I think it will be nice also, it's just a comment to say, for us, South Africa, if we can have such, knowing Arabiensis as our main vector, I think we would really appreciate, so that we make sure that we control whatever local malaria, local ongoing transmission. Thank you.
Yeah, thank you for the comment. And the work has been done in South Africa, in Limpopo, in the Vembe district. It was Prof. Leo Burke who did trials with ivermectin, treating cattle, and they tested it on the local populations there and saw some very nice, effective results. So we are definitely looking at it in the South African context.
Okay, sorry, I forgot. I wanted to add to say, maybe if you can use our local colony.
Yes.
Yeah.
Yeah. Thank you.
Anyone else? Last questions? Okay, thank you.